Hello everyone, welcome to my YouTube channel. Today we will look on distribution of drugs part 2. Part 1 is in the above link, so you can go ahead and check. First is organ tissue size and perfusion rate. We need to know what is perfusion. Perfusion is the passage of blood, a blood substitute or other fluids through the blood vessel or organ or tissue. Okay, and perfusion rate is defined as the volume of blood that flows per unit time per unit volume of tissue. It is expressed in ml per minute per ml, where the unit time is denoted in minutes. Distribution constant follows first order reaction. Okay, so and it is denoted by kb. So with equation we can see that Kb is equal to perfusion rate upon Kt by B, where Kt by B is tissue blood partition coefficient. Next is permeability. Permeability is the ease of molecules across the biological membrane, or ease with which the ions or molecules pass through the pore of the channel. Permeability is rate limited only if the drug is ionic, polar or water-loving, or if there is a physiological barrier which restricts the diffusion of drugs into the cell. Perfusion is rate limited only if the drug is lipophilic, which is like opposite of what is mentioned above, and if the membrane across the drug diffusing is highly permeable as of capillaries and muscles. The thing to note is greater blood flow, faster will be the diffusion. The next is extent of distribution of drugs. So the distribution of drugs depends on the size of the tissue. So increased size of the tissue, increase will be the distribution rate. And second is tissue blood partition coefficient. So it means like if there is a greater partition coefficient, greater will be the diffusion rate. So for drugs like thiopental, okay, which diffuses in both organs, that is the brain and the adipose tissue. So as you know, brain is a highly perfused organ, so naturally the distribution is rapid, while adipose tissue comparatively to the brain is a lower perfused organ, so the distribution is slow. But in this case it is obtained that compared to the brain, adipose tissue is higher in volume. Therefore, the distribution, or there is a greater affinity for the drug, so it approaches equilibrium faster compared to the brain. The other factors which affect the distribution of drugs are age, pregnancy, obesity, diet, disease state and drug interaction. So moving with the first is age. So age, it is classified according to the age groups. So if you see the total body water, it is greater in infants. So the distribution naturally increases, while the fat content is higher in infants and elderly. So there is possibility that highly lipid soluble drugs or acid drugs distribute faster. The third is skeletal muscles, which is lesser in infants and elderly. In accordance to the organ composition, the blood-brain barrier, denoted as BBB, is poorly developed in infants, and also the myelin content is low. The cerebral flow in infants is high, so there is a greater penetration of drugs in infants. Fifth is the plasma protein content. Okay, the albumin content is low in infants and elderly, so drugs which are more likely to bond by plasma proteins and distribute tends to lower. The second one is pregnancy. So in when pregnancy, the volume of distribution increases. This is because the growth of uterus and placenta increases. As I told you, the size of the organ also matters. In pregnancy, the plasma and extracellular volume increases while the albumin content decreases. So there is lesser distribution of drug via the plasma proteins. Third is obesity. A obese person has high adipose tissue content. Okay, so there is increase of lipophilic drug distribution while the perfusion is low. So therefore, it becomes difficult for penetration. And third is high fatty acid levels. Hence, the binding character of acidic drug increases. Next is the disease state. So there is an alteration, okay, in the tissue pH, albumin, and drug binding protein concentration in a disease, in a person who has a particular disease. So the sometimes it can hinder the distribution of drug. In some cases, it is reduced, leading to due to perfusion of organs or tissue. And in some cases, it is altered due to permeability. Like for example, in meningitis and encephalitis, okay, it is a disorder related to the brain. Okay, so the blood-brain barrier becomes more permeable. So the drugs which normally do not cross, lipophilic drugs which, polar drugs or hydrophilic drugs which normally doesn't cross, okay, they it becomes easy for them to gain entry into the brain, like penicillin G and ampicillin. The last one is drug interaction. So what is drug interaction? It is a interaction or a reaction between two or more drugs or between a drug and a food, beverage or supplement. So taking a drug while having certain medical conditions can also cause a drug interaction. So a drug interaction can also affect and also cause unwanted side effects in many cases if it is not taken properly. Our next topic is volume of distribution. A volume of distribution is an important topic it is denoted by VD, and it is also known as apparent volume of distribution. Okay, like literally volume of dilution. So it is the theoretical volume, okay, that will be necessary to contain the total amount of administered drug at the same concentration that is observed in the blood plasma. So in simpler words, okay, it is the ratio of the amount of drug, okay, that is in the body to the concentration of drug that is measured in the blood plasma and unbound in interstitial fluid. It is denoted as VD. Okay, so VD is equal to X upon C. Like I said, X is the amount of drug in the body, and C is the plasma drug concentration, that is how much drug is concentrated in the blood plasma. Therefore, the dose required to give a certain plasma concentration can be determined if the VD for the drug is known. Okay, the VD is not a physiological value, it is more a reflection of how a drug will distribute throughout the body depending on its physiochemical properties. So VD is obtained by checking out solubility, charge, size, etc. So the unit for volume of distribution is typically reported in liters. As body composition changes with age, VD decreases. So as a person grows older, like we have seen before, the volume of distribution tends to lower down. So the drug gets poorly distributed in the body. So VD is also denoted as VD is equal to VP plus VT in the bracket FU upon FUT, where VP is the plasma volume, VT is the apparent tissue volume, FU is the fraction unbound in the plasma, and FUT is the fraction unbound in the tissue. So now our next topic is specific tracers or markers. So tracers are used in medical field quite a lot. Okay, so they are used in medicines and the complex organic molecules, okay, specifically chosen for affinity of the organ under examination. So these molecules are marked by the presence of a radioactive nucleus, and they are mostly injected intravenously. Intravenously meaning by IV route, that is through direct administration into the blood. By injection, you can say in simpler terms. So the volume of each physiological compartment can be determined by the use of specific tracers or markers. The plasma volume can be determined by the use of high molecular weight or substances that are totally bound to the plasma albumin. For example, higher molecular weight dyes such as Evans blue, Indocin green, and I-131 albumin. So with this tracer molecules, you can determine how much plasma volume reaches that particular compartment also. So when a drug is given IV, that is intravenously, they remain confined to the plasma, that is through the blood. The main route is the blood. So the cellular fluid or the extra fluid volume can be determined by substances that easily penetrate the capillary membrane and distribute towards the extracellular fluid, but do not cross the cell membrane, like sodium, chlorine, bromine, cyanide, thiocyanide, and sulfates, and also ions and inulin, mannitol, and raffinose. These are examples of the ions or biological substances which do not cross the cell membrane easily. So here we can see this is a table, okay, which gives information of the physiological fluid compartment, okay, and the markers that are used, okay. So before that we need to know what is the total body volume, total body water volume, okay. So it is determined by the use of substances that distribute equally in all water compartments of the body. So all the water compartments in the body as a whole, okay, both intracellularly and extracellularly, account for the total body water volume. Example: heavy water, the formula D2O, tritated water, H2O, HTO, and lipid soluble substances such as antipyrine. These are the examples. The intracellular fluid volume is determined as the difference between the total body water and the extracellular fluid volume. So there's an equation, okay, to find the ICF, that is intracellular fluid volume, okay. We can it is found by the difference between TWD, that is TBW, that is total body water, minus ECF, that is extracellular fluid volume. So the intracellular fluid volume, including those of blood cells, is approximately 27 liters in our body. So for plasma, the markers used are Evans blue, indocyanine green, I-131 albumin, etc. And for erythrocytes, it is CR51. For extracellular fluid, the markers used are non-metabolizable saccharides like raffinose, inulin, mannitol, and radioisotopes of selected ions, that is sodium, chlorine, bromine, and sulfate. And for total body water, it is deuterium oxide or antipyrine. So towards the last topic of our syllabus, the methods that are used for studying drug distribution pattern. So the methods are microdialysis, matrix assisted laser desorption, ionization, mass spectrophotometry imaging, okay. Short form is MALDI-MSI. And the third one is autoradiography, and fourth is positron-emission tomography. So in microdialysis, the technique involves insertion of fine probes into the tissue of the living body, mostly into the fluid species such as the CSF stands for cerebrospinal fluid. Okay, that is mostly present in the backbone region. Okay, which is connected to the cerebrum of our body. The cerebrum is the brain. So, and the other extracellular fluids were possible. So the probe consists of two concentric tubes. Okay, like you can see in the diagram, there is two concentric tubes and a semi-permeable membrane that is separating them in the middle. Okay, positioned such that the artificial dialysis fluid can be slowly infused through the probe and pass the membrane. Okay, so unbound drug molecules in the tissues surrounding the probe diffuse into the flowing disilate, which is then collected for analysis. Okay, so you can see at the end of the diagram there is a disilate. Okay, okay, and you can see it is in the brain cerebral fluid. Okay, so there's a perfusate at the top. So unbound molecules, okay, they mostly the probe diffuses into the flowing disilate, so which is then collected for analysis. The limitation for this is it is invasive in nature. The next is MALDI-MSI. Okay, or it is used to study drug localization within microenvironmental tissue compartment. So there are thin sections of animal tissues are exposed to pharmaceutical drugs. Okay, either by spotting or by submerging. So then this medical, you can see this mass spectrophotometry imaging method is carried out. Okay, so on the tissue, so you can localize the drug distribution pattern. So this is not a clear image, but you can see there is a stepwise procedure which is carried out, in which the sample is loaded, okay, and it is applied to the MALDI matrix. Okay, that is like an image preparation. Okay, so the sample attaches. So meaning there's an attachment of the sample slide to a MALDI adapter. Okay, so then the it is loaded into an instrument. Okay, here it is given as MALDI TOF MSI. Okay, and then a spectrum or a graph sort of spectra is found. Okay, so where they query further analysis, and the data is analyzed in 2D image shape. So to find out how efficiently the drug is distributed, and to find out to carry out further studies on the distribution patterns. The third is autoradiography. In this approach, the radioactively labeled substance under investigation is administered to test animals, usually mice or rats, which are killed at a suitable time interval, and frozen sections of the tissues or of the whole body is prepared. Okay, so then an image of the radiation is obtained by placing the sections next to a photographic emulsion. So a frozen sections of the tissues are taken, okay, and you place it next to a photographic emulsion. So after that the whole body section is used, and the radioactivity is specifically localized in highly perfused organs. Okay, example lungs, liver, and also brain. Okay, so the image reveals the drug distribution. So comparison with the normal photograph of the slice is used to identify which tissue contains the radioactivity. So densitometry can be used to qualify the relative amount of radioactivity. The technique has been used to show highly specific localization of drugs in tissue. And the last is positron emission tomography. So here a synthetic radioisotopes, okay, with the atomic mass less than naturally occurring stable isotopes having shelf life of 200 of 2 110 minutes is usually taken, okay, and emits positron that interact with the electrons to emit gamma radiation that can be detected outside the body. So the isotopes are generated in a cyclotron and incorporated into the drug molecules immediately before the administrative of the drug. Then this instrument PET scanning permits the production of images of live organism including human beings. I hope you like this presentation. Thank you. Do like, share and subscribe.